Okay, and last talk of the morning before lunch is Anna Lakunina from the Allen Institute, who's going to tell us about Neuropixels Opto.
Thank you, Nick. Yeah, so I'm Anna. I'm a scientist at the Allen Institute, and I'm going to be presenting another next-gen Neuropixels probe type called the Neuropixels Opto, which has been designed to optimize optogenetics experiments. So I have a couple of obligatory slides about why you should want to do optogenetics experiments, because we want to study how behaviors are generated by the interactions of many different brain regions and also the interactions of many different cell types. Um, we have some examples here of like D1 and D2 cells and nucleus accumbens affecting appetite and hunting behavior in mice. But basically, over the past couple of years, there's been an explosion in works kind of generating these massive brain-wide cell type taxonomies across the entire both rodent cortex and subcortical regions. But currently, we don't actually know what these cell types are doing and how they interact with each other and how they generate these behaviors. And so the way to bridge this gap in knowledge is to perform recordings where we can both identify and manipulate these cell types to understand what effect they have on behaviors. And the way that you would do that, currently, the best way to do that is optogenetics. But optogenetic experiments are not done as often as perhaps we would like them to be done because they can be very challenging experiments. First of all, because you need transgenic animals in order to do this, which is like a whole different can of worms that we're not going to get into in this presentation. But also, even once you have the necessary transgenes in the animal, it can be very technically challenging to perform this experiment. I have a couple of examples of common pitfalls here. So for instance, you can just not be recording and delivering light in the same place, which will lead to your experiment not working. If you're doing something like delivering light via a fiber, the fiber can cause a lot of damage to surrounding brain regions as you insert them, which regions that might be important to, whatever behavior that you're studying. And also a big one is just the fact that due to the photoelectric effect, as Andrew talked about, when you're shining a light on these recording sites, you generate these big electrical artifacts that can really mess with the quality of your recording. And so in order to try to address some of these challenges, we are presenting the Neuropixels Opto, currently still in the prototyping stage. So these are some prototype devices that we have from IMEC. Do not be alarmed by the size of them. They will get smaller in the future, but these are designed to be perhaps not quite as plug and play as the ultra probes. They do require, specialized head stages, specialized base stations, but they are made to be more or less compatible with the current NeuroPixels ecosystem where, you can use like the same National Instruments hardware or other hardware that you're currently using for data acquisition, and then visual for acquisition and visualization. They are compatible with both Spike GLX and OpenEFIS. So the NeuroPixels Opto has a site. The size and the spacing of the recording sites is about the same as the NeuroPixels 1.0, with the exception that instead of that checkerboard pattern that you see in the 1.0 probes, they are oriented in two columns of recording sites on the two edges of the shank, with the space in the center being occupied by these emitters, which emit either blue or red light from 14 different emission sites down the length of the shank. So these probes have in addition to all the other hardware components that the regular 1.0 probes have, they have an additional layer that contains photonic waveguides that guides the light down the length of the shank and out a specific emitter that can be activated using the OpenEFIS or Spike GLX GUI. Or more commonly, the way that we use it is we use the OpenEFIS API to send signals to OpenEFIS to open specific emission sites in order to present light stimulus from a specific emitter. And you can see here that you can turn on and off individual emission sites during the experiment to present a very localized light stimulus. So when we characterize the sort of the light emission from these different sites, you can see that there's they kind of exit the emission site in a cone that pretty well covers the area of the brain tissue that is surrounding this shank. So every emission site covers an area that's um, with good power, maybe like roughly 200 cubic microns around the emission site, and we find that gives pretty good coverage of all of the brain tissue that is near these recording sites. So, and one of the big upsides of these Neuropixels Opto probes, partially because the required power is much lower than if you were using a fiber or a collimator to deliver light from the outside, and partially because these emitters are actually they're on the shank, so the light is directed away from the recording sites. We see a huge reduction in these photoelectric artifacts compared to other ways of light presentation. So in this experiment, I was presenting both surface stimulation with a collimator and stimulation from the Neuropixels Opto. So these are 20 trials that are averaged together and median subtracted. And you can see that even with this sort of level of post-processing, you see these significant photoelectric artifacts caused by the surface stimulation, but these artifacts are virtually eliminated in the Neuropixels Opto probe, which cleans up the data pretty significantly.
Is this in a brain or is this in a?
This is in a brain. Yeah.
Do you have an example of the spikes that responds to the input?
Yes. Okay. Here's an example of spikes. Okay. This is an experiment performed by one of our collaborators in UCL, showing. So here we're expressing a redshifted excitatory opsin in excitatory cells in visual cortex. So you see units that are responsive both to visual stimulus and to a surface laser. But you can very precisely stimulate like very localized populations of neurons just by turning on these different emitters. So for instance, with the surface stimulation, we're just kind of stimulating anything that the laser can reach simultaneously. Whereas with by using the emitters on the Neuropixels optoprobe, first of all, you might notice that you are getting some responses that we weren't getting with the surface stimulation. So the Neuropixels opto, it can also help reach neurons that would have been otherwise difficult to reach with these other stimulation paradigms. But also that you can very precisely control like which units are being stimulated just by adjusting like where on the shank you are presenting the stimulation protocol. For our experiments at the Allen Institute, we've mostly been focusing on using these probes for optotagging. So this is an example experiment where we were recording from the dorsal striatum, we're tagging D1 neurons with Kocher and D2 neurons with Carmine. And again, because these Neuropixels optoprobes have these both red and blue emission sites, we can tag these two cell populations simultaneously. So this is an example recording where we have here are 5 example units that we tagged that we identified as either a D2 cell or a D1 cell based on their ability to follow a train of blue laser pulses. And you can see that as before, there is a specific emitter that each cell is driven by, to the emitter that is closest to that neuron on the shank.
They're far red. These are like far red because the blue can activate the red sensor.
That's right, yeah. So in this case, yes, because Carmine is also sensitive to blue light, we can tell them apart because Kocher is not sensitive to red light. So in this case, we could identify D2 cells as if the neuron responds to red light at all, then we would say that this is a neuron that's expressing Carmine. But if it responds to blue light and only blue light, then this is a neuron that's expressing Kocher. This works for us in this case because there's at least in dorsal striatum, there's very little overlap between cells expressing D1, the D1 receptor and the D2 receptor. This could be like a bit of a problem if you're like looking for overlapping cell populations, in which case it would be difficult to tell apart if this is a neuron that's expressing carmine or co-expressing coacher and carmine. So you might want to more carefully select your red opsin in that case, like maybe try something like RS carmine. But that can just be a problem in general with redshifted opsins because they often have that long tail into the blue light.
Also, like, how do you manage co-relief of human and do like, do you have a technique to measure if they are co-relating?
For this experiment, no, we were mostly just working off of the fact that in dorsal striatum, there's very little co-expression of this. Like I said, for other experiments where this would be important, you might want to either better, like be more careful when selecting the red opsin or just not look at overlapping cell populations. Okay, and then same experiment. So those are just 5 example units that we selected, but because of the density of the emission sites, and because the emission sites are in the same location as the recording probe, in this experiment, we were actually able to assign a cell type to the majority of units that we recorded in the striatum. So we are avoiding like one of those major pitfalls of like, how do we know that the light is in the same place as the recording location? It definitely is because the emission sites are on the shank, and that lets us be far more efficient for these optotagging experiments. If we look at the spatial distribution of tagged units versus untagged units, so here we are comparing, we're plotting every unit compared to the emitter that is driving spiking, which for the untagged units here is basically just a uniform distribution because, it's just random which emission site ends up driving it. But you can see here that for tagged neurons, the distribution is biased towards being below the emitter that's driving it, which kind of makes sense if we go back and look at sort of the light spread. It is sort of angled down on the shank relative to the surrounding recording sites. But if we instead compare the distribution to just the nearest emitter, what we find is that the distribution of tagged units is actually pretty uniform compared to the untagged units. The untagged units have like a bit of a larger tail kind of near the edges. So like for some of these units that are kind of further away from the shank. So we might be missing some tagged units that are like particularly far away from the shank that are like right on the edge of being able to be picked up by the recording sites. But for the most part, it seems like having these emitters be 100 microns apart gives us pretty good coverage over the entire like volume of brain that we are able to pick up signals from. And this may also change in the future with improvements to the emission sites to allow us to emit more light power from them as well. There are some current limitations with the Neuropixels Opto. Again, these are prototype devices, and we're still working with IMEC to kind of improve these devices and make them better for future users. In the current demonstrator devices, there is much higher power output from the red sites than from the blue sites currently, and also that there's quite a decent amount of light leakage from unselected blue sites. So in this case, for instance, we are trying to present light only from this bottom blue emitter, but you can still see that there's a decent amount of light leakage from the other emission sites as well. This is a problem that we've already discussed with IMEC, and even like the next iteration might be able to fix this by having 2 separate layers of waveguides for the red and the blue sites. Another issue that we've noticed that might just be laser related and not necessarily Neuropixels Opto related is there's some instability if the red light output is kept on for too long. Another kind of major limitation is just the fact that these current probes only have 14 emission sites per color, and they're 100 microns apart. So the emission sites only span the bottom 1400 microns of the shank, which kind of limits like where you are able to do these optogenetic experiments. Another current downfall of the NP Opto is that it is quite large. Here I have a model of it in pinpoint compared to the mouse brain. And here I have two of them. Currently, they're just in a cleaning solution, but this is that cone that we used at the Allen Institute as a sort of tail guard. So this is the diameter of the well that we implanted into these mice, and you can see just having two Neuropixels Optos together takes up like the entirety of that space in there. So currently, the maximum number of Neuropixels Opto probes that we can insert into the mouse brain is 2, and only if they're very specifically like oriented relative to each other. But hopefully these are limitations that we hope to solve in the future. So for instance, reducing the form factor of the Neuropixels Opto to be about the same as the current 1.0 should greatly improve our ability to stick a bunch of these bad boys into the same brain. Increase the number of emission sites to 44 per color so that we can greatly increase the span on the shank that can be covered by these light emission sites. And also in the future, integrate the light source into the probe base. Currently, every Neuropixels Opto probe needs to have 2 external laser sources hooked up to it, a red one and a blue one, which, if you're trying to record with a bunch of these, that can be a little ridiculous. So for the future, it would be nice to have that just integrated as part of the probe. Current timeline, next year, we're hoping to have move into the alpha probes, which will have the smaller form factor and larger amount of emission sites. Beta probes in 2027, testing the integrated light source. And then if all goes well, hopefully by 2028, the Neuropixels Opto probes will be available for purchase from IMEC, and everyone can start using them for their optogenetics experiments. So thank you to everyone in AIND who has helped with all these experiments, as well as all of our collaborators working on this Neuropixels Opto project, and happy to take questions at lunch.
You did a great job catching them up on time.
Oh, great.
Why don't we just take maybe
Sure.
some questions if people have them, and then we'll.
Yeah.
So how do you manage the temperature depending activities? Then like, because the laser is gonna cause some activity, right? And that is gonna cause some LFPs.
Yeah, the emission sites are quite small, and the power that we deliver out of them is also like relatively low. So for instance, in this experiment, we're delivering 60 microwatts of laser power. So I would expect that the heating that we get from like tissue heating that we get from the Neuropixels Opto is much less than even something like surface stimulation or having like sticking a fiber in, because those we generally use much higher laser powers, like in the milliwatts. We haven't noticed any significant effects of heating from using these Neuropixels Opto probes. That said, we have been using them at relatively low light powers. Like in my experiments, the highest power that I've used is 300 microwatts. It's possible that like if you try to bump that up even more to do something like not just optotagging, but like trying to do causal manipulations on cells around, it's possible that it could lead to some heating. But again, like I would expect that the heating would be less than what we would be currently seeing from regular optogenetics experiments with like a tapered fiber. Yes.
What is the I guess laser gating limit? Like, is it such that you can only deliver square pulse, or rammed or task?
Yeah, so you can deliver arbitrary waveforms from these. Basically, because these are hooked up to currently they are hooked up to an external laser, you can just deliver whatever like analog waveform you want to the laser, and that's what will be emitted. I assume that once we have integrated light source, that will still be the case, that you can just present arbitrary waveforms.
Yeah, I think they were asking us about that
in
our design principles that we want to do, and so
yeah,
they're going to design it in some way controllably.
When you say integrated light source, does that mean transitioning to like a micro LED, or it's still going to be laser.
Yeah, what they're planning to try, I mean, this is, in the future, but what they're planning, it's not LED based. This is, you can tell this is like waveguide based that lights coupled in. There's something called a micro optical mesh, which produces a small, I think, coherent light source in a tiny little chip that they can put on the. That's how.
Yeah.
How do they expect the price tag to compare to their other?
That I do not know.
Yeah, and that's definitely more expensive than standard Neuropixels, but hopefully not exorbitant. So
Right now, they're literally priceless since we mentioned.
Yeah.
You don't want to break one.
Yeah.
Okay, one better.
Yeah, the sort of the analysis pipelines that I've used have been basically more or less the same as what I've used previously for other regular optotagging experiments. If anything, they're a little bit, they might be even a little bit more effective here because you're removing like some of the noise issues that are caused by like external stimulation. Yeah, I don't necessarily foresee the Neuropixels Opto needing like specific analysis pipelines other than just, to other than just to take into account that there's like 40 will be 44 different emission sites and finding units that are responsive to just specific sites. But the responses that we see that are being driven by these emission sites are very similar to the responses that you would see being driven by external light stimulation. In fact, we actually have some experiments that I didn't show in this presentation where I guess, yeah, this group did something similar to what I did, which is present both surface stimulation and Neuropixels opto stimulation in the same experiment. And again, like the responses that you see that are being driven by the surface laser are pretty similar to the responses being driven by the emitter. And if anything, you get more units being driven by the emitter because they are in a better place to drive these responses compared to like a surface laser.
Say really quick because I did say that in the last one. Okay.
So I guess you cannot use color for tracking the electrons.
You can.
You can?
Yes, and that's we do, as with the other Neuropixels experiments, we do dip the probes in dye to track them afterwards in histology. The dyes don't seem to significantly affect the light emission. I always test my probes. I always test the light emission sites on the probes before I insert them into the mouse because the current demonstrators are very finicky in terms of like how you plug in the light cable and stuff like that. And it's very sad to like have this whole mouse set up and stuff and, have the probe in the brain and then the site switching isn't working or something. But I can tell you visually, you can see with your naked eye, like the emission sites lighting up on the probe when you test it, even with even after dipping the probes in dye. And all of these responses were collected in probes that were dipped in dye. So it doesn't seem to substantially affect the ability for these emission sites to drive cell responses.
So that's an improvement to the ones that we are currently measuring by.
Oh, really? Okay.
Interesting. Okay. Will you and you both be around at do lunch?
Yeah.
Okay. So any further questions for Andrew or you can have lunch. So now it's time for lunch. Let's thank Anna one more time.